Welcome to the Explainer. Today we're looking at the memory paradox, surviving the synapse wipe. You know, for decades, the physical architecture of mammalian memory was basically considered a closed case, but that case has just been blown completely wide open by a massive, rule-breaking breakthrough in neuroscience. We literally used to think we understood exactly how brains stored and held on to memories, but, well, a stunning new discovery has just upended everything we thought we knew.
Okay, let's dive into this. What if losing your brain's connections didn't actually mean losing your memories? Just imagine your brain is a giant, complex city grid, right? And suddenly, half the roads just vanish overnight. You'd naturally assume there's no way you could reach your destination anymore. And honestly, that is exactly how neuroscientists used to view memory loss. But what if the destination is still sitting there completely safe, just waiting for the roads to be rebuilt?
To really appreciate why this new research is so mind-blowing, we first have to look at engram neurons. Basically, when you experience something, like a sight, a sound, maybe a specific smell, these specialized cells jump right into action. They encode this fresh information into a memory by forming new junctions, which we call synapses, and by strengthening the ones that are already there. Historically, we assumed our memories were stored kind of like a computer's rigid hardware wiring. The thought was literally that the physical structure was the memory.
And that brings us to what we can now confidently call the old rule. For the longest time, the strict scientific consensus was that memories only persist as long as the specific pattern of synapses on those engram neurons is perfectly maintained. Scientists even ran experiments using drugs to disrupt those specific synapses, and sure enough, the memories seemed to just vanish. The logic was simple, binary, and honestly pretty brutal. Break the physical connection, and boom, the memory is erased forever.
But then, nature threw us a massive curveball. Enter researcher Kazumasa Tanaka from the Okinawa Institute of Science and Technology. He noticed a fascinating paradox involving animals that hibernate. You see, during hibernation, brain activity basically drops off a cliff, and the brain undergoes this massive physical remodeling. Yet when these natural hibernators wake up in the spring, they seamlessly remember their friends and exactly where they hid their food. I mean, how is that even possible if their brain's wiring just went completely offline and shifted around for months? So Tanaka decided to use this natural phenomenon as a really clever way to poke and prod at the established rules of the brain.
To solve this mystery, Tanaka and his team set up this brilliant experiment using mice. Now, mice don't naturally hibernate, so the team had to safely induce it to study the effects. Here's how they did it. First, they placed the mice in a chamber smelling distinctly of alcohol and gave them a mild foot shock, which instantly creates a strong, fearful memory associated directly with that scent. Step 2. They induced an artificial state of hibernation for 2 days using metabolism-slowing drugs and a pitch-black environment. Finally, the waiting game. They brought them out of hibernation, waited 5 full days, and then tested their recall.
So what actually happened? Remember, the brains of these hibernating mice had essentially shut down and physically remodeled themselves over those 2 days of forced sleep. Based on the rigid old rules of neuroscience, that fearful memory should have been completely scrambled, if not wiped clean entirely. But 5 days later, when placed back into a chamber with that distinct alcohol scent, the mice froze in fear. Despite all that intense physical brain remodeling, the memory survived perfectly intact. No way, right?
Now, what's really interesting about this slide is the sheer scale of the biological destruction that occurred while they slept. It is absolutely astounding. Brain scans of the mice's hippocampus, which is a brain region totally crucial for memory, revealed that a massive 55% of the synapses within those engram neurons had completely disappeared during hibernation. By all traditional logic, deleting more than half of your physical hard drive should permanently corrupt the file. Yet, the mice clearly remembered the shock. So, how on earth did the brain pull off this magic trick?
So, the crucial point is that not all synapses are created equal. The researchers discovered 2 completely distinct types at play here, serving very different functions. First, you have what they call clustered synapses. These are packed closely together on the surface of the neurons. These clusters stayed entirely intact during hibernation. You can kind of think of them as a highly secure, damage-proof vault where the actual memory data is stored. Then you have the non-clustered synapses. These are the ones that disappeared during the deep sleep. They act as the access point, or the key to the vault. The vault itself was never destroyed. The brain just temporarily lost the keys while it was sleeping.
And this brilliantly illustrates the ultimate resilience of the mammalian brain. The researchers found that those non-clustered synapses, the lost keys, didn't just vanish forever. Astonishingly, they reappeared within a single day after the mice woke up from their artificial hibernation. The brain literally physically rebuilt the specific access routes needed to retrieve the stored memory from inside that protected cluster vault. The hardware temporarily disconnected, sure, but the core software was perfectly preserved the whole time.
This finding is just an incredible leap forward. Steve Ramirez, a neuroscientist at Boston University who reviewed the study, summed up the magnitude of this shift perfectly. He said, "Memories may be harder to break than we thought." And he hit the nail on the head. This is a profound paradigm shift in science. It completely redefines what is necessary and sufficient to preserve a memory. We are moving away from a fragile, wire-dependent view of the brain to an incredibly robust, dynamic one.
Let's move on and see how this builds into something even bigger. Why does this matter for humans? Well, because our brains store information in a very similar way. The molecular pathways these mice use to protect their clustered synapses and to quickly rebuild their non-clustered ones, well, they likely exist in our heads too. The team is now exploring exactly how these pathways work. If we can eventually target them with drugs, it offers genuine, real-world hope for treating severe cognitive decline. In conditions like amnesia or Alzheimer's disease, the tragic assumption has always been that the memories themselves were destroyed. But this research suggests the vault might still be completely intact. We just need to help the brain find the keys again.
Which leaves us with a final, truly awe-inspiring thought to ponder. If our lost memories, our forgotten childhood moments, the faces we struggle to recall, if they're actually still hiding safely within the clustered vaults of our minds, how long until we find the key to unlock them? Thank you so much for joining me for this explainer. Keep questioning everything, and I'll catch you next time.